understand worlds. So when we look at something, such as this cup of water, we're able to imagine what to do with it. We can look at any object and we know what to do with it. We can imagine picking it up, throwing it around. We can imagine what sounds it makes, how it might feel. And this imaginative power of our brains is one of the most incredible things that we can do. So yeah, got just this little drawing of a dude imagining drinking this cup of water and imagining the sound that it makes and how it might feel. And this is a power that we currently don't know how to give to things that we make. So any robot that exists, any AI that exists, the Siri on your phone, does not have this ability to just perceive something, collect data from the world, and then imagine what to do with it. So this, we believe, is a really important power of the brain. And we think that this is something fundamental and something fundamentally different about living things versus non-living things. So in order to understand this power a little better, what we do is we watch animals as they move through interesting worlds. And my lab primarily works with rats. Also, we do a bit of human movement studies, and now, because of me, we also study cuttlefish, but primarily we work with rats. And so what we do is we put rats into interesting environments, and they're interesting because there's either food or water for them to go find. And we want to see how they behave in the presence of these interesting objects. And then something else that we do is we make these worlds change suddenly. And when these worlds change, the animals look surprised, and they look like they're trying to figure out what happened. So once after this happens, after the world changes, we notice that the animals move very differently once they have changed their expectation about the world. So once they realize that the world can change, we see this realization reflected in the way that they move and in their behavior. So by studying these animals' brains both before and after they change what they expect, we hope to better understand the brain's power to understand, and not only understand, but to simulate and to imagine the world that we live in. So when we're trying to look closely at the brain, we're faced with a really, really big problem. And I don't know if you're going to be able to read it, but it says 84 billion neurons, which is our current best guess for how many neurons exist in the brain. And so when we're trying to look at the brain at work, right now, given the tools that we have, we can only really listen to a couple of them at a time, even though there are many of them working together simultaneously. So something else that my lab does is we build better ways to look at or listen to more cells at the same time so we can better understand what they're working on. Now, the animals we usually study, such as rats and humans, we tend to hide our brains deep inside our bodies. But in order to study behavior and to understand it in its full context, we want to be studying animals that are moving freely and that haven't had any invasive work done to them. So when we're studying rats and humans, it's difficult to look at a brain while it's working inside of a living, moving animal. Luckily, for me, this is somewhat less true about cephalopods and about cuttlefish, because instead of packing 80 to 90% of their neurons into a brain like we do, more than 60% of cuttlefish neurons are out in the body. And as other people have been mentioning throughout the last few days, there's a strong indication that what the cuttlefish sees and perceives in its environment is reflected in a very sensitive way in the cuttlefish skin, in the action of some special cells called chromatophores. So this is why I started studying cuttlefish, because I thought, hey, this would be a really great way to non-invasively watch neurons in action. Non-invasively is really important to me because, as we were discussing yesterday, we are trying to push towards being more conscious and aware of the needs of other animals. And also from an evolutionary standpoint, we know that we are not entirely separate from other animals. We're all shades of each other. And so any pain and suffering that we can conceive of a human feeling, some shade of that, some version of that is possible in other animals. And so just as a creative constraint to myself, when I started my PhD, I decided that anything we don't find ethical to do in humans, I won't do to any other species. Not that I don't think invasive work is important sometimes and justified sometimes, but I think the bar has gotten really low. And so I just want to push us to think about that. So as I mentioned before, the neurons in the cuttlefish skin, they control special cells called chromatophores. And I know that we have a variety of backgrounds here, so just to do a quick background on chromatophores. They're like a tiny bag of color that is held nice and tight, and they have muscles holding onto them from all sides. And each muscle is wrapped around by 3 or 4 neurons. And when these muscles are relaxed, the bag of color is held tight and can't be seen. However, when the neurons tell the muscles to pull, the bag of color can grow up to 5 times larger than it was before, and the skin in that area changes color. Sorry. I'm just waiting for my drawing to catch up to where I'm talking. So yeah, those are all the neurons wrapped around the muscles, and that's how a chromatophore looks when it is contracted. So the cuttlefish skin has about 20 million chromatophores, and each chromatophore is controlled by 20 or more neurons. So this means just in the skin alone, there are about 100 million neurons that are controlling how a cuttlefish appears to other animals. So this is a system that is really, really good, because we all acknowledge that we think cuttlefish camouflage is amazing. So it's coordinating this huge number of units in order to change how, sorry, I'm going to pause real quick, change how a cuttlefish, its form, its color, and its feel. So given all of this is why I started studying cuttlefish, coming from a behavioral systems neuroscience perspective. And so I believe that looking carefully at the cuttlefish skin can tell us how, or can allow me to watch neurons in action without cutting open any part of the animal. So to test this idea, I was lucky enough to collaborate with Hanlon at Woods Hole in the US, and we built a water box, and we called it the Cuttle Shuttle. That's my advisor's name, I can't take credit for that. And at one end, we created a safe place for it to relax, and at the other end, we offered it some food. This was actually just some pieces of frozen cocktail shrimp that I put at the end of a skewer that was controlled by an Arduino. So we kept the walls and the floor, everything very gray and boring, because we wanted to see what the skin looks like when the cuttlefish isn't busy trying to hide. And so I worked with 6 animals, although one animal really didn't want to do anything inside of the box, so I'm only presenting data from 5 animals. And so in order to get the food to enter the water, the animal needed to go to the home side of the box, and once it did, the food would enter the water about once a minute. And you're going to see this guy's going to notice, and it's going to turn and position itself. So They get to eat for free 2 days out of the week. 1 day out of the week they fast, and the other 4 days they have to catch whatever food they want in the box. So we're going to see now the cuttlefish is going to go over and catch it. And yesterday, Jennifer, you mentioned that there's this pattern that shows up only when they're attacking. And I got really excited when you said that because I've been seeing this in these animals. And I wasn't sure as of yet whether this was only because these particular animals have this quirk, because they're all laboratory raised, they live in the same environment, and they're also old. They're 15 months old and sexually immature. But anyway, so now this view is, I've gone and cropped the video and aligned it so that the mantle is always on the right, in the same place. And you can see again, there's this high spatial frequency pattern that shows up right after they throw their tentacles. And it lasts for a really long time when they make a catch. So this is a matrix now of all the catches that this animal did. The upper left corner is the first tentacle shot catch that it ever made, and the bottom right is the last one that it ever made. And so now, this is the moment when it goes ballistic, and you can see across all instances, there's this beautiful high spatial frequency pattern that shows up and persists. Now if you look at all the misses, this high spatial frequency pattern will flash very briefly, but then it'll go away. So you'll see now, we'll wait for the video to go, and then tentacles now go ballistic. And by ballistic I mean the tentacles are pushed out, and then they kind of toss them. And that's when I think someone mentioned yesterday, this is the open loop. That's where they lose control, so to speak. And this was something that, sorry, I'm going to pause, that to humans, to us, any time I showed these videos to a person, it was immediately obvious. I'm like, oh yeah, that thing is happening. That's a phenomenon, and it's a thing that's happening. But to get a computer to be able to detect this automatically was super, super difficult. We tried a lot of different measures. We tried taking average luminance, and taking that measure. We tried taking the mantle image and mapping those pixel values into frequency space to see, because we have a lot of acuity in our hearing, and I just thought maybe, I don't know, maybe we can hear when the body pattern is going super fractally. That didn't quite work out either. Finally, we decided to run it through this machine vision algorithm called CANI, which is based on the human retina. What it does is it looks for high contrast edges. And all we did was count the number of high contrast edges, and you can see in the upper left corner here, when it makes that shot and when that high spatial frequency pattern appears, the graph just shoots up. And so, unfortunately, we discovered that last week, so I don't have any plots or quantitative data to show to you, but hopefully I'll have all of that soon. And I just wanted to, just so that you can see that it's not just in one animal, it was across all of our animals. These are more matrix ethograms, so all the catches are at the top and all the misses are at the bottom. And again, you'll see that they go for the catch, and that high spatial frequency pattern flashes briefly and goes away in the bottom images, but persists in the top images. So that was Dora. This is now Scar, who had a couple more tentacle shot instances. And again, you're going to see that the images at the top persist with the high spatial frequency pattern, and the ones at the bottom, it'll quickly go away. And they kind of go back to something more or less imitating the gray floor, or they go back to something else that I noticed that I was really excited by, Jennifer, when you mentioned personalities. I also started noticing not only behavioral personalities, but also the pattern that they tended to wear. Once they got used to the box and they realized, oh, there's nothing really here for us to camouflage to, they all kind of developed their own default pattern that they would tend towards most of the time while they were waiting around or just exploring the box. It's still unclear to me what exactly they were doing, but for instance, this guy, Plato, really liked keeping those long stripes down its back. And again, it's a slightly different flavor of high spatial frequency, but you still see it showing up in the catches, and in the misses, it's already gone. And then, so this one, there wasn't really a good way for me to summarize the behavioral personalities in videos, but so this is my attempt to summarize the body pattern personalities by showing you, so each of those matrix videos, I've now stacked them on top of each other, and I've had the images add up so that only things that exist in all of the images show up very strongly. And so this was a way of trying to show you that the patterns that show up most often, their default pattern, tended to be slightly different across individuals, but tended to be similar enough within individuals that the images stacked on top of each other reinforce that. So to carry these ideas further, I'm now collaborating at the Brighton Sea Life Center with Carrie Perkins and Daniel Osorio to take more videos of cuttlefish freely moving in enriching environments. And what I'm trying to do now is I'm trying to come up with situations, games really, where I'm presenting cuttlefish with a food reward, but I'm showing them things that are seemingly impossible. So what I'm looking for is, can I surprise a cuttlefish? Now, why surprise? Surprise is actually an abstract concept that is a behavioral manifestation of when our expectations are broken, when something happens that we didn't expect, and we're like, wait, why did that happen? And so expectations is also an abstract concept to unpack, and expectations is, expectations is how we have modeled the world around us, and anytime that model is broken, that's when we're surprised. So this was an idea that I got when I learned about Jean Piaget and all the work that he did with development of intelligence in human children and in infants. And I realized he did a lot of work with pre-verbal humans. And I realized, wait, other animals, when all we can do is study their behavior, we can treat them like pre-verbal humans. So if there's an entire literature on infant cognition studying what we think is going on inside of the minds of babies, just based on the way that they're moving, just based on what we can see in their behavior, then maybe this same philosophy can apply to the study of behavior in other animals. This just to thank my lab, the Kampff lab, and also Roger Hanlon. This is a fortuitous picture that we got when Roger visited Lisbon. And these dot videos at the bottom here, this is all of my lab mates walking, and this is also just to show you, my lab works very heavily with video data, and this is almost the entirety of what we do. We analyze videos all the time. So what we started to do is we just started throwing all of our data sets onto YouTube, just because it's a free place where anyone can access videos, where we can just say, hey, if you check out our videos on our YouTube channel, then you can see all the science that we've been working on. So this is how we've been attempting to kind of open up our lab. And then in case anyone is interested, here's just, so this is our lab website. The second one is my personal website where I have an open lab notebook. It's, again, another creative constraint that I've given myself is that I'm not going to publish in any journal that has a paywall. So I'm actually quite interested in Frontiers because it's an open access journal, so I'll check it out. So I've been publishing all of my work, writing everything up in my open lab notebook. And like I said, all of our data is on our YouTube channel, so if you want to access all of the experimental sessions that I did at Woods Hole, that's that first link. I've also uploaded the cropped and aligned videos that I used for the matrices. I'm going to be uploading all the videos that I used for this talk, and if you ever want to get in touch about anything, that's my email. So thank you very much.